assistant professor in the Department of Biochemistry and Biophysics at Oregon State University. And he's a key member of the GC for All team as the associate director of the GC for All grant, and has been instrumental in all things GCE related at Oregon. However, all of the things that he's most well versed in coding post-translational modifications into proteins to advance the structural biology of their therapeutic protein complexes. So Rick received his bachelor's in chemistry from Middlebury College in Vermont. He then went on to get his PhD at Oregon State University under the guidance of Andy Karplus and Dan Arp. He then did a postdoc at Cornell University in Ithaca with Holger Sonderman before returning to OSU as an assistant professor in 2016. So he's been prolific in publishing and also being funded along the way, including now as part of the GC for All team. He's received numerous awards for his work in structural biology. And with that, take it away, Rick.
All right, thanks, John. Can you see my screen okay?
Good to go.
Cool. All right, well, thanks, John, for the intro. And thank you all for joining us here today for our first International GCE webinar. And thank you, Virginia, for that really wonderful talk. You've certainly set the bar high for the webinar series. So as John mentioned, I'm a structural biologist by training. And when I started my career here at OSU, I became interested in studying molecular mechanisms by which post-translational modifications regulate protein function. And it was clear when I entered into this project area that GCE was going to be the way to tackle these problems, but we needed ways to improve and optimize the technologies to do what we needed. And so after a few years of doing this, I think we're now at a place where we are starting to learn some exciting new biology. And so today, I want to tell you a little bit about some of that work and how these efforts have helped us to receive funding by the NIH to establish the first National Center for GCE Development, which we call the GCE for All Research Center. And so this center is led by our director, Ryan Mell. And we are devoted to making genetic code expansion technologies easy to use and accessible. And so as many of you know, a lot of researchers out there who would love to use genetic code expansion to advance their research, they've either tried and failed, or they never tried at all because they heard it was too hard. And so the mission of the center just fundamentally is to make high impact GCE tools, as well as protocols that are robust and enough to be easily used in any lab. And so in the center, we're focused in two primary areas. The first one is led by our director, Ryan Mell, and that's for establishing and optimizing GCE technologies for encoding tetrazine amino acids, which can be used for site-specific and rapid bioorthogonal ligation chemistries. So we can attach things like fluorescent probes, spin labels, polymers, et cetera, that are conjugated to transcycle octene molecules. And these ligations can be done both in vitro and also in live cells. And so that's one project area in the center. The other one is the one that I lead, which is for developing and optimizing genetic code expansion technologies to encode post-translational modifications, as well as biophysical probes, such as the direct encoding of fluorescent amino acids, as Virginia was just talking about, such as acridone, as well as pi-cation probes, and also fluorinated NMR probes. And so what I want to tell you today is a little bit about some of the developments that we are making here in the Center for encoding particularly phosphorylated amino acids, phosphoserine and non-hydrolyzable phosphoserine. So these two amino acids here. And so as a lot of you, or some of you at least probably know, several years ago, Jason Chin's group published what is probably considered the gold standard for encoding phosphoserine into proteins using genetic code expansion. And when we got that system in our hands in our lab in the center, it obviously has worked really well for us, been able to make a lot of phosphorylated proteins, but we wanted to sort of expand the versatility of the technology by creating a truncation-free expression system using a healthy expression host, as well as a host that permitted homogeneous phosphoserine incorporation. And so what I mean by truncation-free expression is, as many of you know, when we encode non-canonical amino acids, we aminoacylate our tRNA with our non-canonical amino acid, and then this tRNA suppresses a UAG stop codon to encode our protein with a non-canonical amino acid. But as the cells are doing this, they're in constant competition with the release factor 1, which is the cell's protein that would naturally terminate translation at UAG stop codons. And what this does is it means that in addition to making our full-length protein with amino acid in it, we also co-express truncated protein. And this can be seen quite easily on a SDS page gel. We can see we make full-length protein containing non-canonical amino acid, but also get a lot of truncated protein built up. And so to get around that, we've kind of, what is often done historically is we just append a C-terminal purification tag to our proteins, and that way we can just purify out full-length protein, and in a sense we can just kind of ignore the truncated protein. But this is sometimes a problem for people who recombinantly express proteins in E. coli. You can't always just express them with C-terminal purification tags, and so we wanted to make it more versatile than that. And so what folks have done, a variety of different labs, they've engineered cells, strains of E. coli that lack release factor 1. And so this means now that our aminoacylated tRNA can come in and suppress a UAG codon without the worry of forming prematurely truncated peptide. And that way, it gives us the ability to use N-terminal affinity or solubility tags to purify our protein. So again, this in principle sort of expands our toolkit and strategies by which we can express proteins. Now, the one thing that we often don't acknowledge in the field when we use these release factor 1 strains is that while our tRNA is no longer in competition with release factor 1, what it is in competition with are natural tRNAs that will suppress the UAG codon through what we call near-cognate suppression. And what happens is that if our GCE system is not very efficient, which it turns out most of them that we've experienced are not, what happens is the near-cognate suppressing tRNA will come in and then incorporate a natural amino acid into our protein. And so instead of getting a mixture of full-length and truncated peptide, we can get a mixture of full-length protein with our non-canonical and full-length protein with natural amino acids, which is of course not what we want in genetic code expansion. And so we wanted to develop a release factor 1 deficient cell line that was compatible with phosphoserine incorporation, where we didn't get truncated protein and also didn't get a lot of notable amounts of near-cognate suppression. So in 2015, Sakamoto's group, through a series of really clever engineering tricks, were able to develop a release factor 1 deficient strain of E. coli that they called B95 cells. And these cells, surprisingly, were able to grow at the same rate as the parent strain BL21DE3s. So, as opposed to a lot of prior RF1 knockout strains that grow quite slowly and often don't grow to very high densities. And so we were excited when we saw this development of this RF1 knockout strain, and we asked if we could effectively make it compatible with phosphoserine incorporation. And the way that was commonly done is we note that phosphoserine, the free amino acid, is a natural precursor to serine in the biosynthesis of serine. And so what is commonly done is we'll knock out the SERB gene here, and this causes the cell to build up large quantities of intracellular phosphoserine so that then can feed the genetic code expansion machinery. So we knocked out SERB in this B95 cell line, and now the doubling time was still about 40 minutes, so still a reasonably healthy growing strain of E. coli. And now, so we had an RF1 deficient cell line that was also compatible with phosphoserine GCE. So how well did this system work? Well, for that, we referred back to our classical fluorescent reporter protein, GFP, with 1 or 2 stop codons. And when we express these proteins, what we find is that we can about double the yields of singly incorporated phosphoserine into GFP using our RF1 deficient cell line compared to the parent cell line that contained RF1. So we get about double the yields for single incorporation and about triple the yields for double encoding of phosphoserine into GFP. So we're getting better yields with this knockout strain. Now, in terms of fidelity, where we accurately encoding phosphoserine, and for that we can purify these proteins and run them on what we call a PhosTag gel. So PhosTag gels are really a fantastic tool. They're just like SDS page gels, but they have an additive in them called PhosTag. And what this additive does is it will transiently interact with the phosphate group of phosphoproteins, and it will cause those phosphorylated proteins to migrate slower compared to their non-phosphorylated counterparts. And so we can see here in our purified proteins, we have wild type GFP produced, and then our singly phosphorylated GFP produced either with the RF1 containing cell line or the new B95 RF1 knockout cell line, we get clean incorporation of single phosphoserine and near clean incorporation with 2 phosphoserines. And so we're getting better efficiency, we're getting good fidelity with phosphoserine incorporation. Now, in terms of truncated protein, we turn to a modified fluorescent reporter protein where we have an N-terminal purification tag. And so when we express singly, doubly, or triply phosphorylated reporter protein, we see that when we express the protein in an RF1 containing cell line, we get lots of co-purified truncated protein, but in the RF1 knockout, you can see that we have mitigated the presence of any co-purified truncated protein. And of course, when we run phos-tag gel on the purified proteins, we can see we get clean single, double, and triple incorporation of phosphoserine. And so what this means is that now we've sort of, while conceptually this is a somewhat simple advance, practically what it has done is it's really expanded our ability to express phosphorylated proteins in E. coli. And the overall system, the platform, I think, is a lot more versatile. As part of our mission in the center, we're now working to get this system publicly available to everybody. So the Chin Lab has deposited the machinery plasmid on Adgene. We've deposited GFP control protein, control plasmids, and we're about to put the B95 cell line on Adgene. And we're in about 2 weeks, we're about to publish optimized expression protocol in the peer-reviewed journal Bioprotocol. So this whole kit, we hope, is going to be become much more accessible and expand for the community. Now, moving on to non-hydrolyzable phosphoserine. Now, in 2015, when the Chin group published their efficient GCE machinery for phosphoserine, they effectively just adopted the same system for non-hydrolyzable machinery. So when we got interested in encoding non-hydrolyzable phosphoserine, there already was an efficient genetic code expansion machinery for this. So we got really excited about it. But unfortunately, what we ran into was that although the machinery was really good, our ability to actually produce proteins with non-hydrolyzable phosphoserine was not what we thought it was. And so that is demonstrated here. We turn back to our GFP reporters. When we use this system and we add to the media exogenously the free amino acid to the media, we only got a slight bump in total protein production compared to cultures that lack the amino acid. So only about 1 or 2 milligrams of GFP protein from a liter of culture. And our attempts to do double encoding for the non-hydrolyzable amino acid, we didn't get really any protein. So while the yields were very, very low, we were able to purify out a little bit of this protein. And you can see here, when we run it on a phos-tag gel, we have wild type protein, we have protein with a single phosphoserine, and then the protein we purify with this system, we do in fact get encoding of the non-hydrolyzable phosphoserine. And so one thing to note, of course, is that this non-hydrolyzable protein with non-hydrolyzable phosphoserine, it migrates slower on a phos-tag gel compared to the or the equivalent protein with phosphoserine. And so this provides a really convenient way for us to establish whether or not the protein contains phosphoserine or non-hydrolyzable. So the point here again is that the system was totally functional, exactly doing what it was supposed to, but we weren't getting very good yields. And it wasn't scalable. And so we were asking, how can we improve this? And one thing that we noted, so when we do genetic code expansion, most of the amino acids that we add to the media, they're hydrophobic or neutral. And so they diffuse right across the membrane, and that provides enough amino acid inside the cell to then drive the incorporation into proteins. But what I just sort of mentioned earlier is that with the phosphoserine system, right, we modified it so that the cells would produce large amounts of phosphoserine inside of the cell. And so that way the cells produce it inside, and that feeds the GCE machinery. And also when the Chin group developed the phosphothreonine system, they found that they actually had to engineer a biosynthetic pathway to synthesize phosphothreonine inside of the cell to then drive the machinery. And what that effectively means is that charged amino acids, as people have shown, is that charged amino acids, when you supplement them to the media, they just don't get inside the cell very effectively. And so we were thinking that, okay, if we're adding non-hydrolyzable phosphoserine to the media, it's probably doing the same thing. It's just not getting inside the cell. And so our low yields weren't a problem with the GCE machinery itself, but we were thinking that maybe the low protein expression was simply caused by low bioavailability. So how do we fix this? Well, we asked a very simple question, basically the same thing. Can we find a way to actually biosynthesize non-hydrolyzable phosphoserine? And so we went digging into the literature, and we found a paper from 2008 from the Zhao lab at the University of Illinois. And what they did is they were looking at a biosynthetic pathway from a Streptomyces bacteria that would convert phosphoenolpyruvate into this anti-malarial compound shown here through this 8-step biosynthetic pathway. And what we noticed in this is if you look at each of these structures, if you look at compound 6 here that they noticed, they proposed that this was an intermediate in that pathway. And if you think about what the structure of non-hydrolyzable phosphoserine is, this is exactly the same molecule. And so we are excited about this. And so we just simply asked, can we take these first 5 enzymes for D, C, A, B, and E, express them in E. coli, and allow us to convert phosphoenolpyruvate into the non-hydrolyzable phosphoserine to then drive the synthesis of proteins with genetic code expansion. So that's what we did. We cloned these 5 genes, and in front of them, we put 5 different P7 promoter variants with different efficiencies of transcription. We made all combinations of 5 promoters in front of 5 genes, so it's about 3,100 different variations of the pathway. We coupled this pathway with cells that contained the machinery for non-hydrolyzable phosphoserine incorporation, and then of course our GFP reporter with a tag site. And so the idea is that if there is a functional variation of this pathway in the library, it would feed the machinery and then suppress the tag site and GFP and cause the cells to fluoresce. So we were really excited when we did this, and we found that indeed cells containing the tag codon were starting to fluoresce, and they did at different levels depending on what pathway that they received. And so we screened a large number of these different clones and pathways, and we were really excited to find one in particular that when we used the biosynthetic pathway, we got about a 50-fold improvement in total protein production compared to cells that were synthesizing GFP when the amino acid was added to the media. And we were able to encode, in fact, 2 instances of non-hydrolyzable, whereas we could not when amino acid was added to the media. And it works comparably well to the phosphoserine system, about half or 2/3 as efficiently, depending on the protein in the site. But of course, just because we're getting protein, are we actually encoding the correct amino acid? And for that, sort of the best way to establish correct encoding is by mass spec. And we are able to show that when we mass spec these GFP proteins, we found that indeed the protein that was produced with non-hydrolyzable phosphoserine was exactly 2 Daltons less than that of the same protein produced with phosphoserine as a result of the switch from oxygen 18 to the 16 Dalton methylene group. And if we encoded 2 instances of these amino acids, the difference would of course go to 4 Daltons, and that's what we saw. So we're really excited about this. This means that we are now getting efficient protein production with non-hydrolyzable phosphoserine, and because the cells were biosynthesizing this amino acid from essential metabolite, the system is now scalable. But as we were bringing this system online, what we were hearing from people in the community was there was a certain amount of skepticism as to whether this amino acid truly recapitulated the functional effects of phosphoserine. And so now that we had the ability to produce proteins with both phosphoserine and non-hydrolyzable phosphoserine, it allowed us to start answering some of these questions about whether it truly is a functional mimic. And so the first question we asked is, could we substitute phosphoserine with non-hydrolyzable phosphoserine to form a phosphorylation-dependent complex? And for that, we turned to 1433, which is shown here in green. It's a dimer. 1433 is a well-conserved hub protein found in all eukaryotes. It binds to over 2,000 different proteins inside the cell, but it does so when the partner protein it binds to, its clients, are phosphorylated at specific sites. And so it's a phosphorylation-dependent complexation between 1433 and its clients. So we asked, could we substitute phosphoserine here in these clients with non-hydrolyzable phosphoserine? And so as a proof of concept, we chose a client heat shock protein B6, which is a well-studied protein client for 1433. We co-expressed heat shock protein B6 with 1433, where the 1433 is untagged. And when we encoded, of course, just the natural serine and purified out to heat shock protein, we do not co-purify 1433. If we included the phosphomimetic aspartate, it did not form a complex of 1433, but if we encoded phosphoserine or non-hydrolyzable phosphoserine, both of these allowed us to pull out 1433 from the cell extracts and confirm that they were in fact a complex. By phos tag gel, you can see we were getting good encoding of phosphoserine and the non-hydrolyzable phosphoserine. And so we took these complexes that we had purified and asked, how stable are they? What is their dissociation constant? And so by equilibrium analytical ultracentrifugation, we measured the dissociation constant of the two complexes and showed that they both had statistically indistinguishable KDs of around 100 nanomolar. And so from this, we were excited to conclude that, in fact, at least in the case of 1433, non-hydrolyzable phosphoserine, in fact, does recapitulate the functional effects of phosphoserine. So while forming complexes is one function of protein phosphorylation, another function is to disrupt protein complexes. And so we wanted to ask, could we use non-hydrolyzable phosphoserine to do that? And for that, we turned to the same protein, 1433. So as I mentioned, it is in fact a dimer. And what's known is that under conditions of oxidative stress, 1433 itself will become phosphorylated. And it does so at this position, serine 58. Serine 58 is located at the dimer interface of 1433, and it causes what is naturally a dimer to then dissociate into individual monomers. And this happens again during periods of oxidative stress, and then this somehow causes the cells to lead towards apoptosis. And it's not known how this process leads to apoptosis. It's also not known whether by monomerizing 1433, what does that do to its ability to bind clients? Do they get released? Do they still bind? None of these things are known because people have not been able to actually physically produce this material, this phosphorylated monomeric 1433. And so we asked the question, can we use non-hydrolyzable phosphoserine to monomerize 1433? So we expressed 1433 with serine at site 58 with the phosphomimetic glutamate at position 58, and also with phosphoserine and the non-hydrolyzable phosphoserine using our PermaPhos technology. And so we purify these proteins and use size exclusion chromatography coupled to multi-angle light scattering to analyze the oligomeric status of these different proteins. We found the wild type was in fact a dimer as expected. Interestingly, when we used the phosphomimetic glutamate, it also was a dimer, indicating that the glutamate was not mimicking phosphorylation. When we encoded phosphoserine, it was a stable monomer. And when we encoded non-hydrolyzable phosphoserine, we were excited to see that it was recapitulating the monomerization effect of phosphorylation. And so this was exciting for us because not only did we show that it could recapitulate the effects of phosphorylation using stably phosphorylated protein, but now that we had this material in hand, that we actually could use this to start answering some biological questions. And so we decided, well, how does monomerization alter the interactome of 1433? And so for that, we did a classical set of pull-down assays where we took lysates from HEC293 cells, and to those lysates, we added wild type 1433, or monomeric 1433 that either had authentic phosphoserine or the stably phosphorylated form with non-hydrolyzable phosphoserine. So we added these to cell lysates. We found clients would bind to these different proteins. We would retrieve them, wash the resins, and elute any interacting proteins and quantify them by mass spectrometry. Now, one thing to note, of course, is that when we added the monomeric 1433 with phosphoserine, it just got hydrolyzed really quickly, even in the presence of phosphatase inhibitors, right back to wild type. As you can see here in this Western blot, we can see the phos tag where protein with phosphoserine just got hydrolyzed right back to wild type. So we could not, so the data set that we obtained from this was not reliable because it was in fact a mixture of wild type protein and monomeric. But that means that we can compare interactomes using our stably phosphorylated protein with wild type. And so that's shown here in this volcano plot. And in this volcano plot, each individual dot represents a protein that was identified in the two data sets that were pulled down from wild type and monomeric 1433. On the x-axis, we have fold enrichment, and on the y-axis is statistical significance. And what you can see here is that we pulled out about 2,600 different proteins from these pull-down assays, and over about 2,400 of them were found in both, were pulled down in both wild type and monomeric 1433, indicating that basically for about 95% of the interactome, there was no change between these two proteins. So, but we were able to identify 140 new proteins that selectively bound to the monomer, which people have not seen. No one has really identified any proteins that selectively bind to this monomer, but now we have 140 that do. And also there were a select few of 144 proteins that only bound to the wild type protein. Or another way to think about it is these are proteins that are released upon monomerization. So we are really excited about these results because, again, it's the first time the people have shown that we've been able to show that clients still actually bind to the monomeric 1433, but at the same time, there are select populations that will get released upon monomerization and some select population of proteins that will now selectively bind to the monomer. And so what this means is that sort of brief, quick model here is where we have a sort of normal dimeric 1433 under normal cellular conditions. During periods of oxidative stress, 1433 gets monomerized, but 95% of the complexes actually remain intact. But although they're intact, it's still a monomeric 1433. And so the complexes are structurally, dynamically, fundamentally different than what they were under normal cell conditions. And how these function differently is not yet known. But we have about 5% of the clients that do get released, and we can see by the identity of some of these proteins, some of these are pro-apoptotic proteins, and so we can now start to see why monomerization might lead to apoptosis. And we have now a whole slew of proteins that selectively bind to monomeric 1433, and a lot of these are also related to apoptosis and protein degradation. And so we're trying to figure out, how these change in interactome ultimately leads to apoptosis. And so, like I said, we think it's this sort of rearrangement of the interactome that's leading us to apoptosis. And so with that, again, I'll just sort of leave you with, we're excited now to have the center online and running, and we're developing new tools and optimizing these GCE tools. And ultimately, we're really trying to make them accessible for the community. And some of the first two technologies is really creating the phosphoserine tools that are both robust and scalable and have protocols available for the community. And now this PermaPhos technology is enabling us to efficiently and scalably synthesize stably phosphorylated proteins. We can show that it mimics the effects of phosphorylation reasonably well. And we think it's going to be a really exciting tool for uncovering phosphorylation-dependent protein-protein interactions. And so big thanks to our group. All of this work is really a big effort from everybody. Particular thanks to our director, Ryan Mell, who really has led the efforts in establishing the center. And it's a real privilege to work with everybody here. A big shout out to Phil Zhu. He's the graduate student who did a lot of the work that I just showed. And also Stanislaw Stanishewski. He did all the mass spec work that you saw. And so a big thank you to Stan as well. And so with that, I'll be happy to take any questions.
That was great, Rick. Thank you very much. Again, if you raise your hand or you can go into the chat and we can, I'll try to call on you to ask questions. All right, Virginia, would you like to ask a question?
Yeah, thank you. That was a really great talk, and I'm sure a lot of people will be really interested to use the reagents you developed. As we've been talking about, I think that, the field is, one thing the field is thinking about is, first we try to make the proteins in vivo and then purify them out of the cell, but then ultimately we would want to be able to, for example, make a phosphorylated protein in the cell and then study its role in the living cell. What are you guys thinking about making that transition?
Yeah, that's a great question. So Jason Chin has shown that if you encode phosphoserine in a eukaryotic cell, it gets immediately hydrolyzed. There's too many phosphatases to really make it a viable process. But so we are now actively building or adopting this PermaPhos system where we biosynthesize the non-hydrolyzable, and we're now building that in HEK cells as well. So yes, we are going that direction, and I think that's going to be a potentially really powerful technology as well. Yeah, so yeah, we're working on it, is the short answer.
Great. So we have some questions in chat that I'll convey to you, Rick. Is it possible to do similar engineering? Well, this question was just kind of discussed, but is it possible to do similar engineering for mammalian cells to incorporate non-hydrolyzable phosphoSer?
Yeah. So we think it is possible. I mean, the Chin group has done some really exciting work, and they've shown that if you add the amino acid to the media and HEK cells and put in all the appropriate GCE machineries, it can be done. And so they showed proof of concept that it is possible, but we think that with this sort of biosynthetic approach to making the amino acid inside the cell and coupling that with the GCE machinery, we think we should be able to do it in mammalian cells.
And another question from Jan Eric wants to congratulate you on the great talk. And have you made the improvements to phosphothreonine system over the original chin version or even a non-hydrolyzable analog?
Yeah, also good questions. And again, the answer to that is we are definitely working on improving the phosphothreonine system. Because of the popularity of the systems, the phosphoserine system was sort of the first one we've tackled, but we are now also attempting to make the same improvements with the RF1 knockout compatibility and evaluating that as well. As far as a non-hydrolyzable analog, we haven't yet started on that. I'm not aware of a biosynthetic pathway to produce it at this point, but first thing is just, of course, we can synthesize the amino acid and see if we can make it work. So yes, all things on the chopping block for sure.
And I have a question for you, Rick. So have you taken your proteins that you've made recombinantly with non-hydrolyzable phosphorylated amino acid and then delivered them into eukaryotic cells to see how they interrupt biology?
Yeah, we haven't quite gotten there. At this point, the first step was just to see lysates and see if we can make that work. But again, yep, we are. My student is actively working on that at the moment. Yes, I think it's going to be great if you can get that dialed in and get reliable delivery at defined concentrations. I think that could be a very powerful tool.
A couple more questions before we end the session, but this is from David Ewell. How about phosphotyrosine? Are you working on that?
Yeah, so I know. So of course, a system has been published. I think it was from the Schultz group for phosphotyrosine and non-hydrolyzable. I think it's a great proof of concept, but I think there are other folks out there who are working to improve on it now, and that's something where we've paid a lot of attention to. And something I think the center will be interested in working on as well.
And from Martin Hengelsbach, did you consider modifying the non-hydrolyzable phosphoSer with a hydrolyzable but lipophilic protecting group for delivery, kind of like a methyl amino acid that people have used for E. coli?
Yeah, that's a great question. We did not look into that. That's something that I think could be very tractable. So yeah, no, we haven't, but I think that's a great idea. I think the one advantage of biosynthesizing is the scalability, right? And so that if the cells can make it themselves, then you can just grow as much protein and express as much as you want. So that's one of sort of the advantages of the system, as opposed to just improving bioavailability. It's also improving scalability. But I think that's a great idea, and that's something to definitely look into.